This is the number of reactions taking place in the human body per second. That's 37,000 billion. So how are humans able to facilitate this colossal number of reactions? Enzymes are biological catalysts that speed up biochemical reactions. A catalyst is a substance that speeds up a chemical reaction but is not consumed or altered in the process. Today we'll be discussing how enzymes are biological catalysts, the mechanisms of enzymes, examples of enzymes in the human body, extracellular and intracellular enzymes, and the commercial application of enzyme activity. Firstly, enzymes catalyze biochemical reactions by lowering their activation energy. The lower the activation energy, the faster the rate. This is considering that activation energy is the minimum amount of energy required for a reaction to take place. Take a look at the difference in activation energies between the catalyzed and uncatalyzed reaction. The lowering of activation energy is thanks to induced fit. The shape of an enzyme caters specifically to the shape of substrates. A substance to which another substance is applied is what we call a substrate. An enzyme has a specific 3D shape with an active site that fits exactly with a specific substrate. An enzyme may act on one substrate to form two products, or it may join two substrates to form one product. The two reactions that occur using enzymes are the hydrolysis reaction and the condensation reaction. Hydrolysis refers to the process of breaking molecules and compounds with the addition of water, polymers to monomers. Condensation is the opposite. It is the formation of compounds with a byproduct, usually being water, monomers to polymers. An example of hydrolysis is a breakdown of the disaccharide of sucrose, which is made of the monosaccharides, glucose and sucrose. Here we can see the products of the monomers being released. An example of enzymatic condensation is the formation of chains of proteins. Proteins are made out of long amino acids. Long chains of these amino acids code for different types of protein. Fun fact, there are 20 different types of amino acids. So now that we know about what enzymatic reactions are, we can delve into what factors affect the rate of enzyme activity or the rate of reaction. Factors that you should know are pH, temperature, enzyme concentration, and substrate concentration. When the temperature is increased in a reaction, so is the kinetic energy. This increases the speed at which reactants collide, which increases the enzymatic rate of reaction. However, enzymes have an optimum temperature. When the temperature is too high, enzymes can denature. Denaturation of an enzyme or any protein changes the shape of the protein, therefore preventing it from functioning. As we saw earlier, the shape of an enzyme is crucial in binding to the substrate because of the induced fit theory. Secondly, looking at the pH, different enzymes operate at different pH levels, and it is essential for these conditions to be met to avoid denaturation. For instance, the enzymes in your stomach would require a different pH to those in your small intestine. And lastly, the quantity or concentration of enzymes and substrates impacts the rate of reaction. When the concentration of reactants is higher, the chances of successful collision increase, therefore increasing the rate. After a while, the concentration of substrate or enzymes will decrease as it is used up in the reaction, leading to a stable point towards the end of the reaction. Let's go back to that pH example where I mentioned how the optimum pH of enzymes would differ depending on their role. Enzymes have an important role in digestion as this is a large process involving multiple steps, breaking down food components and extracting nutrients, storing energy, and eliminating waste. Examples of enzymes involved include lipase, protease, and amylase. Here is a table showing the respective substrates for each of these enzymes. Lipase having the substrate of lipids, protease catalyzing the breakdown of proteins, and amylase having the substrate of starch. And it's not just the human body. Enzymes are everywhere. Commercial applications of enzymes include proteases in baby food, biological washing powder, and beer and wine production. These applications are almost hidden in everyday life. Protease is applied in baby food since babies cannot yet digest their food well. The protease allows some pre-digestion of the proteins. Biological washing powders contain enzymes for hydrolysis reactions to break down the stains in clothing. And lastly, enzymes are used to speed up the fermentation processes involved in the synthesis of beer and wine. We hope you've enjoyed our video on enzymes. Please feel free to comment questions below and check out our website on science worksheets filled with questions on topics under the BMAT and from this video. Thank you for watching and happy revising. Bye!